I posted a link to the materials in the chat. We're gonna kind of have like 3 sort of breaks in this session. The first part of the session will be focused on some of the standards that we use and that are supported by Tellurium. After we go through, that'll all be in this collaboratory notebook, Reproducible Distribution. So we'll be working out of that first. Then I'll go a little bit through kind of a transition period where we can look at a couple different ways you might actually be programming on your computer if you're not really planning to use Jupyter notebooks in Google Colab. So we can go through that. We'll also go through distributing a study, which will sort of be kind of just lumping in a lot of the different processes that we're doing with this, using the standards for, and the other procedures in Tellurium, and then basically distributing a study to GitHub. So we'll do that. And the last part of the session will be on reproducibility. So we'll kind of go over just a general sort of presentation on that. It won't be any programming, but just some good practices to look out for. And after that, I'll try to leave space at the end to basically ask a lot of questions about anything that's happened throughout the course. And specifically, if you want to try to sort out something about your project, maybe we can have some of those bigger discussions as well. So that'll be at the end of the session. Okay, so we'll start in this collaboratory notebook on reproducible distribution and standards. And the goal of this is really to talk about how we can distribute a complete archive which contains our models and our simulation experiments, which is called a Combine archive. And this can be done with Tellurium. And so we'll go through the standards that basically compose that archive and how we create them with Tellurium. The real utility of using standardized formats is that we can generate a lot of, or we as a community can build tools that support those formats. And this allows our models to be understood and either executed or analyzed on different tools that are created to support the standard, which allows us to basically exchange our models, build upon them, and ultimately just have a more cohesive community that can exchange their models with one another. So in this notebook, there's a table of contents going through the main sections we're going to touch on, which will basically be importing SBML, which is the first standard we'll look at. We've talked a little bit about it throughout the course, but it's what is the primary encoding for models in the systems biology, computational models in the systems biology community. And we'll talk about importing those directly from the BioModels database. And then we'll go on to generating Setml, which is a standard for writing simulation experiments. And finally, we'll talk about creating the Combine archive as a whole. Okay. I also have some links to relevant resources here, which will basically take you through specification and tool support for each of these standards we're talking about. This might be useful to you if you have questions about your specific modeling project and whether some method you use to encode your model could be supported with SBML, and whether there are tools to use to simulate your specific model or visualize, analyze any task you want to perform on your model. So you can look at this SBML tool support. The same is available for Setml tool support. And then some other resources are here that may be relevant. We'll talk about some of these throughout the tutorial. So just consider looking at those afterwards too, if you have additional questions. All right, so we're gonna go ahead and start running this. We really only need to install Tellurium in this notebook because the only packages we're using are Tellurium and SBML throughout or the only non-standard packages we'll be using. And for the purpose of this notebook, we're just gonna be working with one model from the BioModels database, which is this respiratory oscillations in yeast model by Jenna Wolf that we have looked at in some of the previous notebooks I presented. And the first task we're going to go through is how to import and export with Tellurium. And this model is published on the BioModels database. Obviously, Lucian's given you a really strong introduction to the BioModels database. And we're going to be talking about how you can import those directly. So first, let's talk a little bit about what SBML actually is. I know we've kind of touched on it briefly, but we'll just to clarify any doubts about it, it's basically a software data format that's used to describe models in biology. It's a little bit like HTML, but for creating models. So markup languages allow you to separate annotations and documentation about the content you're creating from the content itself using standardized tags that can be read and interpreted by other tools, which means that basically SPML is independent of any particular software tool. And that means it's not limited to any field of the life sciences, and it's also broadly applicable within the biological modeling domain. And many members of the modeling community are developing tools that support the standard so that we can expand how we actually use it. SBML is open and free, and it's widely supported already. And there are libraries like libSBML that can be used to help developers support SBML in their tools. Essentially, the goal is to make software that's able to interpret the tags in the language, encode the model so that it can be used in their own tools. And tools could allow tasks like writing the model, which for example, would be Antimony plus Tellurium allows us to write models and encode them in SPML. Simulating the model, which Roadrunner allows us to do, or visualizing the network that composes the model would be another possible tool. So a tool to support visualization would be another task. So, and while the core features of SBML generally support reaction and process models that we've been talking about throughout this course, there are some extensions that support spatial models, constraint-based models, and rule-based models and logical models as well. So potentially your models might be in one of these different domains, and there could be SBML support. It's definitely it's strongest for those that are reaction-based and process models. Okay. So how do we use SBML? Well, I think we kind of touched on this in the first lecture introducing Tellurium, but we don't write SBML directly. The software that we are using will do it for us. So you can either use the list of material tools that support SBML in the relevant resources, or like we're obviously going to go through how you do use Tellurium to write it. Although we've talked about it a little bit, but we'll go into it a little bit more in depth. So first, we're going to go and import an SBML model from the BioModels database using a URL to that model. Like I said, we're going to be using this wolf model. So in all the examples, the Roadrunner instance for the model is going to be called wolf. And the command to load it from the BioModels database is te load SBML model. And then we're providing it this URL, which is it's taking us directly to the SBML form of the curated model. If we go to the BioModels database, I can show you how that link is actually generated. Let's just look at a random model. We won't look at our model necessarily. Okay, so Lucian took you through how to kind of go through the BioModels database and that there are certain model files. This model file would be is an SBML model that we could potentially import directly. So the way that the link is generated that I'm using in the code is just to basically it's the link that's generated when you open the link in the new tab. So this is generating the link that you would use in the Jupyter notebook. If that's it. So that's just where that's coming from. Okay. All right, so we're loading this SBML model directly from BioModels database, and then I'm just running a simulation as we've done multiple times before, just to show you what the model looks like. So we were able to successfully load the model directly from BioModels. This is really useful. It means that we can directly import other models or published models and use them in our own analyses, or just start programming really simply without having to necessarily download the files directly to our archive. And then it's more reproducible because anybody who has this code can go and grab the same model. All right, so once you have your model imported into your programming environment with Tellurium, you can then also export it to different file formats. So here's just an example of exporting the model. So this will export to whatever my current working directory is. It's creating sorry, just in case, I can't remember if I reran this, but let me make sure I do. Okay. So running this will just, it would, if you're using this in your local machine, it would create a file called this Wolf 2001 respiratory oscillations.xml, which is just an SPML file. It's using the current version. Since I just reset it and I didn't change any of the parameter values, it's the same version I imported from the BioModels database. But using this current tag, if I had changed any of the parameters or if I had run a simulation and not reset the model, then it would export it with the current concentrations for the species and the current parameter values. Okay, so it's a really simple command, just export to SBML. This will get you the file that you can then use. You can do the same thing with Antimony. You can export an Antimony model and save that as well. Here, for the purpose of the rest of the exercises, I wanted us to just take a look at the Antimony. So this is just wolf get current Antimony. I'm printing the Antimony string essentially for the model. And I think we've looked into this a little bit before, but yeah, if you want to take a look at the model, or if you have any questions about it, then Antimony is a great way to do that. We kind of talked about how it's easier to read than SPML, so this will allow us to get an idea of get a sense of the parameters and the species involved and how they're interacting with one another. Okay. All right. So for the sake of some of the analyses that are to come, I had to update this, update the Antimony string for the model to be compatible with the export to the next standard, which will be the simulation experiment description standard. Okay, so here I just basically copied and pasted, copied this model that I printed into into string to generate a new Antimony model. It's essentially the same. I changed the model name, model ID to be wolf here, so it's going to be consistent with the examples for the two standards that are coming down the pike. And then I also removed some of the annotations, which were for the model, which were model identifiers essentially, which just talk about the identity of the model. Basically, it gives you the published model in BioModels and also the PubMed ID for the paper it was published with. So those were removed because they created conflicts with the subsequent export to Setml. All right, so yeah, very short editing. See, this is now the strings ending at end instead of including those annotations about the model. I'm gonna then go ahead and export SPML and Antimony versions of this updated model to my current working directory. I'm gonna need both the Antimony and the SPML to create the Combine archive in the end. So here, or to create it with Tellurium, I'm exporting the Antimony version of this model. It's exporting the version I just updated, even though I didn't really change any of the actual model content, any of the parameters or species. It's exporting it to wolf underscore antimony.txt. This will be an Antimony file, and then I'm also exporting to SPML. So this wolf underscore SPML.xml is going to be the SPML file for the model that I'm going to use. And for the sake of this study, we're going to, just for the sake of the simulation experiments we write, let's just focus on one species in the model. So we're going to work with this KEBI ID 15379 was what it was in the model. It's the species name in the model is Oxy or OXY, which is basically, it's dioxygen. It's a substance that removes electrons from another reactant in a redox reaction. So it's a yeast metabolite in the respiratory process, obviously. Plays into this model of respiratory oscillations. Okay, so just for the sake here, we're going to run a simulation using Roadrunner. So wolf.simulate from time 0 to time 100 with 1,000 time points, only track time and this dioxygen species, and then we'll plot it as we've done. So this is the plot with Roadrunner, and we're going to go ahead and try to recreate this plot using the simulation experiment description language, this Setml, in the rest of the tutorial. Okay, so now we get into Setml. This is our second standard for today. Setml encodes a computer readable exchange format, which includes the information that's required by the minimal information about a simulation experiment guidelines, or MIASE, which is basically these minimum information standards try to kind of, well, it's kind of self-explanatory, I guess. They give you the minimal information that you need to be able to fully specify the experiment you're writing or the model you're writing, so that if somebody else has that information, they'll be able to completely reproduce or they should be able to reproduce the work you did. Okay. Yes. So similar to SBML, this is a technical specification that uses an XML schema, just like SBML. It covers the description of the most frequent types of simulation experiments that are in the systems biology domain, which are primarily time course simulations. Most of what we've been working with have been time course simulations. And ultimately, these documents can specify which experimental data we should use in our experiments, which models to use in the experiment, what simulation we're going to run, and which models we want to run those simulations on, which simulation procedures, which simulation procedures we're going to run on the models, which results we want to track as a result of those simulations and pass to the output, and then how we should display the results that we track in our output. So that's kind of that gives you an idea of what Setml is intended for. Phrased ML, this second term here, is basically the Antimony version of the simulation experiment description language. So it gives us it's a language and a library that can give us a text-based way to read and create the Setml files, because just like SBML, it's not really feasible to write Setml. So Tellurium has functions that allow us to use phrased ML as part of this larger ecosystem that incorporates Antimony and Roadrunner. And ultimately, it allows us to perform the same experiments that we can do with Roadrunner, or most of the same experiments we can do with Roadrunner. And it tries to follow the Setml description format to define models, define simulations, define tasks that are performed using the simulation and the models, and then to generate output. So then we'll go through what each of those mean by looking at kind of the anatomy of the phrased ML string. Okay, so here in this block, we're gonna actually write phrased ML. It's gonna be similar to Antimony. As we said, it's a string format. Sediml models are the first line here is to set the model. So Sediml models are models that can be found with some kind of URI. Basically, you should give some kind of either path or file name that will tell you which SPML model you're going to be using in the simulation experiment. And the syntax for defining the model is here in the comment. So you give it a model ID, and we know we've called our model Wolf in the Antimony string before, so we're using the same model ID here. Then the word model, and then the source model, which is going to be either the file path, the file path, or just the file if you're using the file from your current working directory, which we are. So we have wolf_sediml.xml here, which is the SBML file we just exported before. Okay, the next section then is to actually describe the simulation experiment. So for the simulation experiment, it's basically a description of the type of experiment we want to perform. It describes only the simulation experiment. It doesn't tell you which model we want to use to perform it. So here you don't see any information about the model that you're using. Presumably, you could have multiple experiments on multiple models included. But here we just have the single experiment. Okay, you can do steady state analyses, you can do time course analyses, and then in terms of time course analyses, you can do both deterministic or stochastic simulations, which we've talked about a little bit. Here we're just doing a deterministic simulation. So I've called the syntax for describing the experiment is to have the simulation ID, which we've called det_sim, which is just short for deterministic simulation, and equal sign, the word simulate, and then the simulation type you want to run. We're using uniform, and this is the deterministic simulation that we are most familiar with. It's similar to the r.simulate command in Roadrunner. And we are simulating from time 0 to 500 with 1,000 time steps. So it follows the same sort of format that the simulate command follows. Okay. After you've, so this is defining the experiment itself. The next step is to define a task, which again takes into account not only the experiment that you want to run, but the model that you want to run it on. So it's basically telling you run this simulation on that model. And in this case, our simulation we want to run is the deterministic simulation, and we want to run it on our wolf model. The syntax here is to have a task ID, which we have created as wolf_det_sim. So basically telling you we want to run the deterministic simulation on the wolf model, and then equals the word run, and then the simulation experiment you want to run, the word on, and then the model ID. So yeah, follow this syntax: task ID equals run simulation ID on model ID. Okay, so now you've basically instructed it which simulation experiment to run on which model. And the last step would be to specify what you want the output, or what results you want to track / what you want the output to be. We're going to just do a simple plot, since we've been doing that with Tellurium and libRoadrunner. So the syntax here is to so for the output type here, since we're doing a plot, we're going to tell it plot, and give it, here we're giving it just a title name, and then we're giving it X versus Y axes. There's a lot you can do to change this, so I would recommend looking at the SBML documentation if you want to do something more advanced. But here we're just going to tell it to run. We're just going to tell it to have a really simple title, Wolf et al. Dynamics. Give it a model ID with the BioModels ID, BioModels identifier. And then we're going to tell it to run or to plot time versus the OXY species. So here, time and OXY are obviously the same parameters that were in the Antimony string, with all have the same names. And so we're basically just trying to recreate this. Okay, so that was the anatomy of the SPML string. I guess I'll ask if there's any questions that I've missed.
The only question was like if you could use a URL instead of wolf SPML.xml, and the answer is yes. Mm-hmm. You've changed it, so you want, so that points to a local copy if you use a local file name, but you could use a URL, a general URL if you wanted to.
Yeah, so basically they could, if they wanted to, import directly from the BioModels database.
Right.
Yeah. Okay.
Excuse me, can I ask a question? Mm-hmm. Sorry, my question is kind of very basic, but I didn't understand it. What is the Roadrunner and Tellurium? How you differentiate them? You know, is it a package inside the Tellurium, or how is it like that?
Roadrunner is a library that is included within Tellurium, and Roadrunner executes all the simulations. So it's basically provides like integration capabilities. So it can it basically generates a version of your model that can be simulated. I guess Lucian or Herbert, if you want to comment more on that.
Yeah, that's basically it. The Roadrunner is the engine that drives the simulations, and Tellurium is all of the syntax and everything else, making that easier to use in different contexts, I guess. Yeah, the other general thing I'd say about Setml is just that the reason why you would want Setml is so that you could then import that on some other simulator without having to, so currently we've been writing Python scripts, and Python scripts are great, and they are specific to Tellurium and Roadrunner. So if you wanted to say, give that script to someone else, then they would also be running Tellurium and Roadrunner and sort of they would be able to replicate, like repeat exactly what you did, but they wouldn't be able to sort of test it on a new thing on someone else's simulator, like Copasi, say, without like figuring out what you did and then recreating it in Copasi. So Setml provides a common language of simulation to a variety of simulators. So like Copasi does have at least limited Setml import. So we could write the Setml file, import that to Copasi, and then Copasi would run exactly what we did here in Tellurium in your Python script.
Have you ever heard of Jerkmate? It's just fun.
And so yeah, and eventually, and in fact, you'll find on BioModels, you will find some Setml files there. I don't know if you've seen them sort of passing by on a handful of things, but there is the Setml file that is used to recreate the figures that they've curated.
Mm-hmm.
So again, you can, if you have that Setml file, then you can start directly from, and you know exactly what the simulation conditions are and everything else to recreate the figures that were in the original papers.
Okay. Any other questions about the phrased ML or any of the other import export? Looks like it makes sense. Okay. Okay, so that's pretty much it for this really simple phrased ML string. That's all we need. We've told it which model to use, what experiment we want to run, which model we want to run the experiment on, or the experiment we want to run the simulation on, sorry. The experiment we want to run and which model we want to run it on together in the task, and then the output specification we want for the plotting. All right, so then we actually have to generate the Setml. So this is just a couple of functions here. Yeah, okay. So here I'm just doing a reset. Since I did simulate the model, I'm going to be generating this SBML string to use for setting the referenced SBML. So I just wanted to make sure that I reset it to its original form. Okay, so now I'm going to be passing wolf.get SBML. This is just returning the SBML in a string to the variable wolf _ SBML. And then the main two functions here to convert the phrased ML to the setml is phrasedml.set referenced SBML. So I'm now setting the model reference that we're going to be using here in phrasedml. Okay, so then you give it the location of the file and then also the string. String that I've just.
I can explain a little bit of what that is because it's a little arcane, but basically in the setml or in the phrasedml thing, it is referencing a string wolf _ SML.xml, and the library phrasedml can't necessarily might not be able to find that on your local file system or whatever. So if you need to set reference to SBML, we'll say like, if you want, when you are asked about wolf SBML.xml, here is the string that is that. So you don't have to go to the file system to find it.
Thanks, Lucian. Okay, yep. So that's that line. Then this line here is where I'm actually where you're actually performing the conversion of the phrasedml into setml. This is all happening with functionality in phrasedml, the function convert string, and then I'm passing it the phrasedml string that I've just created here. So this is going to generate a string wolf setml, which I guess I can print that just so you have an idea. It's going to look it's going to look similar to the SBML since it's still an M markup language format, XML format. Okay, so not the most intuitive for us, but very, but well supported by many simulators. So super handy to have this format. Now, so I've just created the setml string. Now I'm going to export the setml to a file using Tellurium. So here I have Tellurium saved file. The name of the file name I want to save it as, and I'm passing it the setml string that I just generated. And then after that, I just want to show you that how Tellurium will then execute that Setml if you just had the file. For example, if you did want to go on BioModels and download one of the Setml files that they have available there for the curated models. So then just running this, I've regenerated this simulation. This is plotting. It's just created the plot that I specified in the SBML file. So it has the title that I specified, and it's plotting time. It's plotting the OXY species against time, 500 time points. Obviously, this doesn't look exactly like the other simulation because we've sampled it differently. We have more time points. We have a longer time course simulation here than we did when we ran it earlier just with Tellurium. So here we were simulating only 2 × 100. But yeah, we were able to recreate this. Well, we were able to create a simulation experiment in the standardized format and execute that with Tellurium. So these are all pretty, once you get to using Tellurium to either to execute files that are existing, the commands are pretty easy. So the anatomy of how to write SBML is really probably the more complicated task here. So as long as everybody is kind of on the same page with that, that's about it. Okay, so the next thing then would be the Combine archive, which also is relatively easy to create once you have the components, which are the SPML model and the Setml simulation experiment. So the Combine archive is essentially just a package that contains all the related standards, and both, like I said, the model and the simulation, so that you can easily exchange an entire modeling study between different software tools. Obviously, if you have the model, then somebody can take the model and can run their own simulations on it. If they have just the simulation experiment, I guess they probably may need the model as well to be able to run that simulation experiment. But with just the model, you might not be able to reproduce the exact results that somebody's created. So having the simulation experiment and the model together is a really handy feature. It's just complete. So Tellurium basically provides a way to write this open modeling exchange format, OMEX, which is then going to be able to which it can basically be edited so that you can change the contents of Combine archives in a human readable way. And this inline OMEX is basically, for our purposes, it's like an Antimony description of the model joined to the SBML experiment description. So we've already created the components that are necessary for creating the Combine archive. All right. So here we're just going to read our Antimony model into a string. So assign it to this variable wolf antimony. And then we already have our wolf SBML, which is the SBML string that we've created. So we're going to then create this inline OMEX string. The syntax here is essentially, like I said, it's joining with a new line. This \n is a new line signature, and it's going to go and join the components that are within this list. So it's going to put a new line between the string for the wolf antimony and wolf SBML. So I'm going to run this. It'll save that to the variable wolf inline OMEX, and then I'm just going to print what the OMEX file looks like. Okay. So here's our antimony model up at the top. It looks like the model that we wrote before. And then below, it's going to have the simulation experiment that we wrote in SBML. So it's literally just joining these two strings. And then to create the Combine archive, then you're just going to use TE export inline OMEX and pass the variable name, which we've just named our string wolf inline OMEX, and the file name that we want to save it as. So that's going to go ahead and export a Combine archive, which now contains our simulation and our model, so that if we give this Combine archive to somebody else, they could go and either use it with Tellurium, they could unpack it, use the component parts, or some software supports the Combine archive entirely. And we're going to go through an example of we're going to go through another example where we'll actually use the Combine archive we've generated once we get to the distribution part. I do have, I don't know if, I mean, it's a really simple exercise if people want to try the import to the BioModels database. That's pretty much what this first exercise is here. So we can do a quick break for that. Otherwise, I can kind of move on to the next point. It's a pretty simple
let's break for that,
task.
I would say.
Okay, we'll give it maybe 5-10 minutes. We can go over it.
You can also answer questions if anyone has anything if you're working on stuff.
Yeah, do feel free to ask questions.
Oh, I just noticed I sent that message to you again.
Right.
I don't look at the thing. Forget.
The way Zoom chat works is that if anyone sends you a private message, then everything you send afterwards is by default just goes to them.
That's
And
probably
so you
safe.
always have to change it back to everyone.
That's probably a safe thing.
Yeah.
You can imagine all sorts of problems.
Share for a sec.
Comparing Kapazi and Tellurium. So Kapazi is a GUI. The biggest difference between Kapazi and Tellurium is that Tellurium is command line Python and Kapazi is a GUI. We have other tools that we have produced in the lab in the past that are also GUIs, but we've sort of moved the sort of to a text-based simulation thing because we found that more useful for our own work. Kapazi and Tellurium sort of date back to the same eras many years ago. So they've both been developed for a long time. Kapazi is, so yeah, it's you get a window, you tell it to import things, you select things from drop-down menus and fill out forms and ask it to simulate things.
Yeah, I guess on the model input specification is via a spreadsheet. So that's
okay,
that's how you specify row by row. You specify everything that way.
Yeah.
But as I said, I suppose on Monday now, I mean, a GUI can only take you so far because you're limited by what the author provided in the GUI. And they can't provide everything. Whereas, I mean, the reason I use this, I mean, and whereas a scripting language, you're basically just limited by your own imagination what you can do. Now, of course, it's easier to get started with a GUI, and it's harder to get started with a scripting language. But I think in the long term, especially if you make this your profession, then a script-based approach is better, I think.
Probably let you do more.
Yeah, if you're a professional. And you notice that the major computational tools, the commercial ones, are all script-based. Mathematica, MATLAB, although they all have a little bit of add-ons for doing GUI stuff as well. But on the whole, they're script-based because you have such enormous flexibility when you have a script.
So there are other things that distinguish the different simulators from each other as far as like emphasis. Like Kapazi is really sort of emphasizes stochastic simulation. It has sort of native things for exporting things in terms of particle number as an example. So it really sort of, it was built with those in mind instead of the continuous ODEs. Just you can use the same SBML file for both, but in one you interpret the ODE as a rate, and the other one you interpret it as a probability of firing, basically, for each of the reactions.
Mind you, having said that, we do support, Roadrunner does have one stochastic simulator inside it.
Yeah. So you can absolutely do stochastic simulation in Roadrunner. It just, as far as what it was originally designed to do, Composite has been doing stochastic stuff for longer and more originally. Like that was what its original purpose was. So some of the, I don't know, extra functions that you might like on that tend to be that. There are other simulators that'll do other sorts of analyses. There's flux balance analysis, which is another whole thing that you can also express in SPML with it's called the FBC package for the flux balance constraints. There's and the other thing is you can sometimes mix and match. Like you can use Tellurium and if you know enough, you can tell it to use the Capacity simulator instead of Roadrunner. If there was some reason that you wanted to do that. At the very base, like if you might be doing something weird and wondering if Delirium or Capacity is actually interpreting your model correctly, so you run it with Roadrunner and then you run it again with Capacity, and then you have a completely different code base running your simulation. You have more assurance that what you're doing is true if they match each other, if they match.
Yeah.
That goes back to repeatability versus replicability versus stuff, which we've talked about a little bit.
Stuff. Technical.
The technical term things.
Repeatability and reproducibility.
Yeah, reproducibility.
Yeah.
So repeatability is generally, we've defined it as doing something again at a different time, at a different machine, at something else. Something I did this once, I want to do it again. It'll do exactly what you did in the same way.
Using the same code, same data, same everything.
Yeah. So if something is reproducible, though, then that means that you are redoing the experiment in a different context, using a different code base, hopefully, using maybe a different set of inputs, or like slightly like a different repeat of something. I don't know.
Yeah, you could have slightly different data.
Slightly different data, slightly different anything, and hopefully that would then if that gives you the same results, if it answers the same question in the same way, then you have reproduced what the original was. So that's what you want in terms of, like, you have a published paper, then I want to do it on my machine. You're not going to have access probably to their exact software setup, but you want to be able to do what they did so you can continue their work. And that's what these standards help with, and Tellurium has really gone full in on reproducibility as far as trying to support these different standards for that.
Okay, we can probably go over the exercise if people did it. It's pretty quick. Was everybody able to do it?
Reply with your thumbs up. There you go. Yay.
Cool. I'll just go through. I mean, it was pretty fast. Really, all you had to do was modify the URL from the original, from the other example, importing directly from BioModels. So this was just BioModels 10, which if you follow the link here, you should be able to follow it to BioModels page for the Kolodenko model and simulate. So pretty similar to what we did before. Okay. I guess.
You want to share your screen, Veronica? Again?
Oh, darn! I wasn't sharing my screen.
Okay.
Okay. Yeah. Sorry. Okay. Can you see it?
Yep.
Yep.
Okay. Yep. So that was the solution. Super fast. Using the code from above. Just change this URL. Okay. Now we're gonna move a little bit into a sort of transition period. I'll do a few. Okay, so transition period, what does that mean? Well, we're going to go through a couple things. We're going to go through a little bit about distribution of a modeling study on GitHub, which will be kind of part of the transition. Transition being that we're moving from using just these Jupyter notebooks that we've all been using this throughout the course to some other options that you might use, so like the programming environment that might be more comfortable to use on your local machine most of the time that you're writing code. So I'll go through a few things, then Herbert and Lucian, if either of you want to share
Sure.
something else, how you would program normally, or
Yeah,
any
okay.
installs or anything, you can
Sure.
go ahead. Really quickly, I think one common programming environment that we'll use sometimes is Spider, and there's a Tellurium version of Spider. So Herbert may go into more about this, but
Yeah. Okay.
there's actually a version of Spider here on Binder, which is basically, it provides a virtual machine that you can run from your browser. So I'm launching right now. Oh, I'll post a link in the chat as well, if you want to access like the same Binder version that I'm doing for this part of the tutorial. Yep, this will allow you to just run this from your browser, so it won't require you to install anything on your computer right now. It might not be a long-term solution, but it'll show you kind of how to use Spider. Okay, so
I want to say what Binder is. What's it for? What is
yeah, it's
Binder?
providing you virtual machines or container. I don't know if it has containers or if they're all virtual machines, I guess, but it provides an environment to work on that has all the software dependencies you would need without actually having to change, like reconfigure your local environment on your computer.
So it saves you having to install an application. You can go and use it through a browser.
Yeah. This is basically like you're looking at a desktop. It's basically a virtual desktop. So yeah, the first step then that we're going to do, because we know we don't have, this Binder is a very generic version. It doesn't have Tellurium already installed. We've been doing pip installs for most of our Jupyter notebooks, and you can do the same here from the command line. Just one line, pip install Tellurium. So this is going to go and grab Tellurium. You might do something similar on your computer if you have pip ready to go. Yeah, otherwise there are other options for downloading Tellurium, but this is probably one of the easier ones. So I'm going to just wait till this finishes installing. It might be almost done.
You need to fix that NumPy version.
Yeah, I don't know what it's doing there.
It's a.
It insists on using 1.19, right?
The current public version of Delirium is I had to pin to a particular version of NumPy because the version, a more up-to-date version of NumPy would crash on Windows or would like had a bug. So I pinned it to the earlier one. The Windows, it was because of a bug in Windows itself.
Yes.
Windows has then been fixed, and so now we can get the.
When we do the next release, we should have do
next
this
release.
thing,
Okay. In fact, it's already in the code base. I think we just need to actually release it.
Okay.
All right. So basically, that pip install just installed Tellurium to this virtual machine that we're working out of. I'm going to go then launch Spider, just like an application, any application you would launch on your desktop. And this is basically just going to give you a really simple interface for programming in Python. I don't need to do a tour today, so. Okay, so these are basically this is just some comments, but what you're writing out of here, this temp.py is going to be a Python script. So we've been working out of the IPYNB files, which were notebooks, but this is just a single script. It's kind of sort of like the equivalent to one of those code blocks in your notebooks that you would run. You can do the same and execute just a single script here. So I'm going to start by importing
Can you get that any
Tellurium.
larger? It's pretty small.
Yeah, it's under tools menu.
In Spider itself?
Spider, yeah, preferences. And then it's
editor.
Maybe
or
not entirely obvious. Oh, there it is.
I don't know if these are going to be, do you think those are the ones? I'll try it, I guess. Okay, cool.
Oh, there you go.
Yay, thanks.
Oh, does that all look fine?
Yeah.
Okay.
Yeah,
so we'll import Tellurium as TE as we've been doing in our notebooks. And then just, I guess we'll do the same type of thing we've been doing. Oops. Which is just to create an antimony string here. I guess we can use the same one that we've been doing, this simple one reaction model, species S1 goes to S2 at a rate of K1 times S1. So mass action kinetics. This is the same example I've used before. I'm sorry. I'm sure Herbert would be much more fast with this stuff.
Yeah, I'm squirming on my seat here watching this.
It's the stage fright. It's the stage fright is why I can't seem to type at this exact moment. I swear my normal program.
You're making everyone else feel much better about themselves because it was like, oh, good, everyone else, other people do the same thing.
Yeah, no, it's bad. Okay, so we'll load the string to Antimony. I mean, sorry, to a Roadrunner instance with Tellurium, simulate it like before. I don't know, 10 points. 10 time steps, 20 data points. Okay, plot it. I think that'll be a
that's it.
Simple script. Hope it works.
This is, you do this once. They want you to confirm that you're using Python, basically. Okay, so now what you do is, yeah, it's okay, it's done.
You have to click on the plots.
See the plot
tab.
button?
Oh yeah,
there you go.
It's been a while since I've used Spider.
Now you can opt to have them pushed out of the console as well if you want. There's a little option you can set.
Yeah, you'd probably have to change something in matplotlib, or would you change it in Spider?
No. There's an option in the top right-hand corner of the plot. Yeah, there, I think, is some setting there.
Well, it actually tells you right there. It says like to make the thing appear in the console, uncheck mute inline plotting under the plots pane menu option. Yeah.
Tells you what to do there if you wanted to.
Okay, well, for now, it's just going to show up here. But that's just a really simple script. Probably will work in this format a lot more than in Jupyter notebooks. Yeah, I don't know, Herbert, if you
the advantage, I would want to say, I'd say one thing. One is you can have as many tabs as you want. So you can create a new one. So do new, top right corner, left corner. Then you can type another model. And so you can have, lots of different models on different tabs. You can cut and paste between edit tabs. I just find it easy to work. And you don't have to worry about whether previous cells have been executed or not, because you just run the entire thing. Although you can have sort of fake cells.
I think you can do you can create cells with this, and then in that case you can kind of walk through them. I'm pretty sure by using
you've done
this. If you had some block in there, then you could just run a current cell. So that's
the other thing. Go back to the first model you had. So if you say select just the plot, or say just select the last 3 lines, and then right click. Right, select and right click.
I don't have right click.
Oh. Oh well. Oh no, I know what you can do. I know what you can do. You select and then go to run on the menu. And then you can, select the cell, run selection or current line F9. See that down there? So you can do that kind of thing. Which can be helpful. Yep. So poor man's cells.
Yep. So this is one option. It's probably one of the simpler options, I guess, for writing scripts. So it might be a good way to start out.
The other thing you have to be careful is with the notepads, if you send it to somebody, they may not be using a notepad at their end. And notepads have special file formats which are not compatible with common Python. So generally, if you want to send a notebook to somebody else who doesn't use notebooks, there's a special save in the Jupyter page. It says save as a Python script, and then you send that to them. There should be one somewhere. Download. It's under download.
Oh yeah, you can download
.py, yeah. And that one will run in any Python environment.
The only problem might be the pip installs. In that case, you might have to
you strip those out.
Have somebody add those to their workflow.
Yeah.
Okay.
Now there's for Windows, we have a full installer on Tellurium Analog Machine, where you can just download and then click and it installs everything you need. and doesn't interfere with any existing Python you have. So
Herbert,
that's,
if you want to show them that
oh yeah,
in a little while,
yeah, I will
that
do then. Yeah,
might be a good, okay.
yeah, okay. Yeah.
Okay. Okay, so that's just, yep, a really fast introduction to Spider. The next one that I'm going to show you, which we'll get into a couple of different things, will be PyCharm, and we'll just kind of go over we'll go over PyCharm and GitHub, and then also just distributing your modeling study, just like one way to distribute your modeling study, and I'll kind of build a little bit on what Lucian taught you about GitHub. Probably his was a lot more in depth, so we'll just go through a few utilities. So I just have a study here. It's on private right now, so this will just be for the purpose of, I might be able to share it later, I guess. But just wanted to show you kind of working with a really simple repository. We're going to pretend that this is some modeling study we wanted to distribute. And I'm working out of PyCharm for this. So this is another development environment. It's kind of like Spider, except it has probably more functionality. Definitely more functionality, I would say. I think it's a little, it's just really handy. I like it specifically because it allows me to directly pull and push to GitHub,
Oh, right.
which is really just like, it makes my workflow a lot easier. I won't go into a lot of detail. It can probably get kind of complicated. I don't probably use it to the full extent of its capabilities, but here I'll just go through the components that I have in here, and we'll talk a little bit about GitHub and GitHub desktop as well. Okay, so in this repository, which we're pretending is a modeling study we've created, I just have two .py files, so two Python scripts. Here we have import model, which is basically just, well, it should really be write model, I guess, and export. But we're just writing a really simple model in Antimony, same one we just used in Spider. And then after we've written that, we're going to export the Antimony and the SPML files. So you'll see those show up over here under the directory. Again, this is just the name for the repository that I have in GitHub that I just showed you. I've basically pulled a copy to my local machine, and I'm working out of the main branch, so I didn't fork it or anything like that. Okay, so I've exported two files to this repository, the Antimony and the SBML file. And then I have another script here that's just simulate.py, which is gonna basically generate the Sediml and then export a Setml file as well, so that I can create and a combine archive. So everything I just showed in the last demonstration in the Jupyter notebook, we're doing the same thing here. So I'm loading this SBML model simple reaction that I exported in the import model file, should be export model. Simulating it just to kind of demonstrate the Roadrunner simulation, and then we're going to go and recreate that same simulation using the Sediml string. So all the syntax here is going to be the same as what we saw in the previous tutorial, running a deterministic simulation from time 0 to 10 for 20 time points, and then specifying the task we want to perform, which is to run this simulation on the simple reaction model, and then doing a simple plot again of time versus species S1 and S2. Okay, and then we're gonna perform the conversion from Sediml to Setml, and we've already gone through the syntax to do that. Exporting to Setml, so we're saving this Setml file, which is the simple reaction _ Setml.xml. And similarly to export a combine archive here, or omex archive, which is going to be simple reaction.omex. Okay, so I've already, well, I guess from just to demonstrate this in PyCharm, I've basically configured this file to be run with just that green arrow. So I'm running the file right now. There's some setting that is off slightly, so the figure is popping up over here. But you can configure the plots as well to show up, so they should show up in line. I think it's a matplotlib setting right now that's not doing that. Okay, but this is the simulation that was generated from the Roadrunner execution here, this simple reaction.plot. All right, so this is went ahead just the first time I ran it, generated this omics archive as well, and the Setml file. So now we have all of our standards here inside of our modeling study. Over on this side tab here in PyCharm, we have project. So this is showing us all the files that we're currently working with. So I can, well, that won't look like anything, but I can look at simple reaction.txt, and this will show me the Antimony file. So you can access all your files from the project tab. Then the other really, I think, useful tab here is going to be the commit tab. So again, I have this configured so that I can use GitHub, or I can push and pull from GitHub directly from PyCharm. So basically, any changes that I've made in my repository are gonna come up in this commit tab, and I can basically then decide if I want to push these to the repository or not. So what I'm going to do is I'll just select the changes to these three main files, the import model, the omics, and the simulate model, and I'll go ahead and write some commit message. And commit and push. So I've
Veronica, does it show you a diff there, or would it? Like, can you? Yeah.
I don't think I've ever seen it on the PyCharm tab. Probably the safest bet let's cancel this push instead. I can commit this. Oops, sorry. I guess it already went through, maybe.
I think it did.
Okay, let's.
And now you're screwed. So.
I'm just gonna add. Okay. Okay. I'll just add like another random simulation here.
So to answer the question in the chat, omics is a zip file, so it is absolutely not human readable. It is a way to bundle all of your stuff together in one place. So inside the omics file will be the SBML and the Setml and anything else you want. So it is a way to have like, here is everything you need. I will send it to you, and then your computer like reads it, and then it gets both the Setml, which tells you how to both the SBML, which is the model, and then the Setml, which tells you what to do with the model.
Mm-hmm. Okay, so I just am adding, sorry, I don't know where, a new simulation. Oh my God. Okay. Yikes. Okay, so now this change is going to show up in my commit, in my new, over in the commit branch here. Simulate model, basically, I've just changed it. So now instead of committing and pushing directly from PyCharm, which maybe I would do if I was extremely confident in my changes, I'll just click the commit. And this is basically gonna, it's not gonna push it yet to GitHub. So I can now kind of show you GitHub desktop. So GitHub desktop is really useful if you're regularly, or at least I find it really easy to use to pull and push to GitHub. Maybe Lucian, you can talk about another way to do that from your local machine.
Sure, I can do a similar demo.
Awesome. Okay, so we'll just kind of go through this. Okay, so I just committed the changes to this simulate model.py, but I didn't push it yet, so they should show up here. And unless I change it, I guess I can. Okay, well, first of all, let's kind of go through, I guess, what's GitHub, this GUI for GitHub desktop. Okay, so starting off, I'm working in this repository that I showed you before online, which is this network modeling summer school modeling study._district, or at least that's what the NMSS stands for. So I basically have any repositories that I'm currently working on, I'll have open, or I'll have up for use in GitHub desktop. So I have multiple repositories here for the SysBio group and for my own personal GitHub account. And I want to work out of this network modeling summer school distrib project, basically. Okay, so I have several changes here. Any changes that have been made since you've last pushed to the main repository are going to be available here for review. As you can see, they're showing the changes that have been made. All of these are just files that have been added. All of them are they're associated with being able to perform these, or basically they're associated with the PyCharm environment. So most of them won't make, they're not like really understandable. Maybe some of the names you won't understand what they are. But these are some changes that can be pushed. I can do that from directly from this platform. So I can write a short summary, and then also I can write a longer description if I want to, and then I would commit that to main if I felt ready to commit. The changes I think that I've made before, oops, I've just pushed them. Okay, the change that I had committed from PyCharm for the for this simulate model file, I just pushed basically to using GitHub Desktop. So you saw there were two. Hopefully you saw there were two changes or two commits here that were waiting to be pushed to the main branch, and so I just pushed those to the main branch. You can look through the history. So this has basically all of the commits that I've made since I created the repository. This initial commit was just creating the repository, which I did online, and all you can see here is a license and a README, both which are pretty standard. Then I had a first commit where I basically executed the .py scripts, and as a result, I had all these standardized formats that I wanted to add to the repository, so I added those. And then here you can start to see some changes. Here you should hopefully see the change that I made in the simulate model. Okay, so here you're seeing the points in the model that I actually updated. Yeah. So this was the last commit that I just pushed it. You can also fetch origin. So I believe it does this periodically. It'll try to fetch the origin. Otherwise, you can just request fetch origin. Sometimes there'll be changes depending if you're working with multiple other people. You might run into changes that have to be merged. So you'll have to resolve those here. Current branch, I'm just working on the main branch, but you could create a new branch here. Pull requests you have available through the GUI as well. So all this can basically be done in the GitHub desktop. A lot of it can also be done directly through PyCharm. Like I showed you, you can do the commits here from the commit branch. You can pull from, I think this will update if I have, if I had pulled through GitHub desktop, I would be able to pull here. And then also down in this lower bar here, right now you're just seeing this run tab. So if I was running one of the files, I could visualize the progress of that run here. There's also this Git tab here. So this is going to show me the same thing you should see here in the history of the GitHub desktop GUI is going to be displayed here in the Git tab. So this is basically showing you all of the commits that have been made. If I forked the repository, then you would get a branch off from the main branch, and you could work along that fork for some time and then potentially merge back to the main branch. And all of that will be displayed kind of visually here, and all the actual text for your commits is going to be displayed here. It tells you who performed the commits and when you made them. So all of that's available there. The other thing that's somewhat useful is just the Python console. So you can directly program from the console. Like you don't necessarily have to create a script if you just want to do something really simple. So I can just import Tellurium. If I just wanted to test my model, then I can run all this code without having necessarily to commit it to a script. So maybe if I was doing something really simple that I just wanted to test, I could do that here. Okay, other useful features, I guess. I find this to-do feature pretty useful. So basically you can create this comment called to do. I don't know. So let's say I wanted to create a different version of the model. I could add this to do comment, and then that'll show up here in this to do tab. So it shows you which files have to dos, and it's just like an easy way if there are some tasks that you have to put off for some time to just add the to do tag to your comments, and then like this will be an easy way to go and check that off. So then once you're done with it, you can just delete the comments and it'll get removed from the to do tab. So I think those are really the ones I find most useful. You also have a terminal here that you can work from. So if I do need to install new packages, like if I want to install tutils, I can do that directly from this terminal line. And this is really useful because in PyCharm, you can configure virtual environments for each of your projects. So I'm using a virtual environment for Python 3.8 in this project. So if I have packages in my primary Python package that I don't on my local machine that aren't in the virtual environment, I would need to add those. And it's easier just to do it from the terminal here, since it's directly working in that virtual environment. Okay, I think that's most of what I have basically.
Veronica, you had one question in the chat, just how to set up the Spider environment on Binder. Was that just pre-set up and you are using it, or is there?
Yeah, I didn't set up. Spider had that on their web page, just a web-based version. I just looked up if there was a web-based version, and Spider had that available.
Okay.
The link to it is in the chat, but yeah, I didn't set that up.
Sorry, how do you log in to the GitHub with the GitHub account here with PyCharm?
Like how did
you originally configure this set up to be connected to GitHub? Yeah.
I honestly am not sure if I can. I mean, I probably just looked. I mean, at the time, I looked up a tutorial of how to link GitHub with my PyCharm, or how to link PyCharm with my GitHub account. It wasn't difficult, but I don't know if I could redo it right now. But there are definitely like good tutorials.
Any of these things that you do once and then never do again is like, I don't remember how I did this.
Yeah, unfortunately, but yeah. I can probably find a good tutorial after today's session and like add that to the Slack, if that would be helpful.
Okay, thank you so much.
Yeah, sorry, I can't answer it. Yeah, I think this is all I have. The idea here is basically just to create so this is a modeling study that I have now on GitHub. Obviously, there's a lot more you could add to your modeling workflow than just having a model and a simulation, but at least you get the model, you get the standardized versions of the model and the simulation, and you have the combined archive here. Something I wanted to show with that, but I might need, I don't know if Lucian or Herbert will be more helpful with this, because you can basically the archive that I created for the simple reaction.omex, you can import that directly into Copasi. I did put installation or download instructions for Copasi in the Jupyter notebook from this session.
Which one? Don't save that.
No, I don't want to say that, sorry. I have, yeah, technically, I actually think I already have it loaded,
Copasi import omex files?
Yeah.
Oh, I didn't know that.
But the thing I don't know how to do is how to actually visualize. The plot is here. Dynamics of simple reaction model is the name of the plot.
First?
Oh yeah, probably.
You have to know that it's a time course.
Time course,
because it hasn't been run yet.
Time course.
There.
Then you have to go output assistant. Then the first one.
Or just run.
No.
You have to do things.
Well, if you want plots.
Well,
nope, just run. Okay.
Yeah.
This is it. This is the simulation. So
0
to time 10. Yeah.
Oh, I didn't know that. That's great.
You do need to know some, like, you definitely need I mean, I've done this before. I just haven't done it frequently enough to remember, but there's a lot of, Coposi has a lot of tutorials available online to show you how to do this, but it does support the Combine archive. So basically what I've just done is I created the Combine archive using Tellurium, created the simulation experiment using SBML, exporting into Setml, and now I can reproduce that same plot that I have in Python in Coposi. So now you've got the same plot, two different simulators. So this is kind of what Herbert was commenting on before, the utility of exporting as standardized formats.
So I should mention that we're pushing, well, most of the community is pushing Combine archives to be the standard way of distributing models. And BioModels, for example, the big repository, if I'm not wrong, Lucian, is moving to all Combine archives. Is that right?
Well, you can already get, if you remember the files directory at BioModels.
Oh, they have them there anyway.
So at least programmatically, you can always get those as an OMX file, as an OMX directory. Like all the files in that thing will then be just bundled up. There'll be
but the new bunch you're making, that'll be
right. I should also mention that, like, don't put too much stock in the SedimL files that you find on BioModels today, because a lot of them don't really work. One of our lab is working with BioModels to clean them all up, and soon they will all work, and there will be more of them, and it will be wonderful. But today it's sort of an exercise in frustration. So
the reason we're going towards OMX files, somebody asked what are they actually? They're just zip files. So if you change the extension to .zip, you'll be able to open it with a zip, with a zip program, and you'll find inside it a whole bunch of files. One of which might be an SPML file, another one might be a Sediml file. There could be other files in there as well, including PDFs, data files. The idea of the archive is to have a container that holds everything you need to run a simulation. Right, everything you need to run a simulation. So, because at the moment, with just an SBML file, you don't know how the model was run. You don't know if it needs any data to run. You don't know, etc. So I think you'll find more and more people hopefully using these Combine archives.
Yeah. Okay, I think that pretty much shows you.
Did you want me to show people about how Git works on my machine, or?
Yeah, that would be awesome.
Sure.
Then I'll show a little bit of Spider, my environment. Sure. Yeah,
that'd be great.
All right. Maybe I'll just share the screen. All right, I'm just share my whole desktop. So here, let me see. So what I have on my machine is called Tortoise Git. I also have Tortoise SVN back from when I used SVN, but now I have Tortoise Git. And what it does is that this is just a normal Windows window. But what Tortoise SVN does is if I'm looking at a archive, it will show me what's changed and what hasn't. So like this is a Git file. Here's my desktop. I have a bunch of things that are, so anything with a check is synced with, like, there's nothing that I have that's different from what last time it checked. And anything with a little exclamation mark, I have changed something that was that I haven't committed yet. So as an example, here's our networking 2021 example. And then in the right-click menu for Windows itself, if I want to sync with the archive, and in fact here, let me do this first. So this is the last time I used the network modeling example. And as you may remember, here's that K0 that's 4.82, which was wrong. I have since accepted someone else's pull request on GitHub itself to fix 4.82 to 48.2, which is what it should be. So at this point, then I want to pull down those changes. So I want to sync with what's with GitHub itself. And I can say pull, and it will pull all the changes. And now if we go back here, it reloads the thing because it changed. And now look, it's 48.2, yay. So now if I want to make more changes here, say I just want to change that to millimole instead of millimole, right? I'll save that. Now, if I go back here, it'll say like, hey, you've changed this file. And then if I want to commit it here, then I select that and it shows me, and here's the place for my message. And what I like about Tortoise in particular is that then it shows me a list of everything. And if I double click on it, it'll highlight the particular changes. So whenever I really try to be good about looking at all the changes that I've made before I commit anything, because invariably, if I don't, then I will have made some change that I forgot about that was just sort of a testing thing. And I didn't actually want to commit it. And then I'll have to make another commit that reverts it or something. So there's my change, milli space mole to millimole. And then I can say demo wording fix, right? And then I can click commit and push and it'll go and upload everything and be good. So now if we go to GitHub, let's see here. We'll go to that example. We can look at my commits and there it is. And then on GitHub, we can also see its version of what the diff was. So that's what I use to interact with Git from my machine. So that's what Veronica was saying. She uses GitHub Desktop, which is another perfectly fine thing. This sort of integrates with the operating system itself, and I've liked it. And I've liked the convenience and the sort of the discipline that it helps me keep of being able to look at all the different changes that I've made in every single commit that I make. And then this is Spider here. This is the collapsed version of things. And if I hit run, then it will, now that it's 48.2, now it's oscillating again. So is there any other questions about that? Oh, here, someone did have a chat thing. Oop, okay. Oh, yeah. All right, I'll stop sharing. So Herbert, you want to show him Spider a little bit?
Yeah, just have a quick look at Spider, what I have.
Yeah.
So this is what mine looks like, and I use it for development and modeling. For development or for very large Python programs, I use Visual Studio Code because it has a proper debugger. So then it becomes really, I mean, when I mean a big project, I mean maybe 20,000 lines of Python or more. And then you need a proper debugging facilities to work your way through it. So that what I like about the Spider, so we distribute this. This is the Windows version, and if I go to the Tellurium web page and then install, and you go down, there's the IDE front end. So the front end Tellurium Spider IDE. This installation with these two here. So this is the Windows one, okay? We have two versions, 3.7 and 3.8. So you pick the 3.8. This is actually a complete solution. So it's a single executable. You just download it, run it on your machine, and it'll include everything you need, including Spider, Jupyter notebooks, Python 3.8, and a bunch of all the common packages like NumPy, SciPy, etc. And it doesn't interfere with any existing Python setup you have. Okay, so we try to make this as easy as possible for Windows people to install Tellurium, and it's just a single click. And we'll be updating this at some point this year. So for the Mac, at the moment, we just rely on Anaconda for the Mac, and then for Linux, you can do pip install and use whatever IDE you like on Linux. Okay, so that's what we have for Windows. And so when you get it, you will get something like, if I bring this up, maybe, where is it? Yeah, you get Spider 3.8. You also get the Jupyter notebooks and Jupyter Lab for free if you want to use Jupyter notebooks on your local machine. So that's quite handy. The other thing it'll do when I do new, it immediately comes up with a template for me. Okay, and you can modify this template however you want, but I can immediately go and start typing in a model. Okay, so this is the one Veronica did, but I'm just doing it a little bit faster.
Shade.
And if I run that, I get the same thing that Veronica got. Okay, so it's very convenient. The other convenient thing is here, the Windows version has a built-in plugin. So if I go to File, it actually has an Open SBML file option. So if I click that, and then I go and find an SPML file, SPML models. Here we go. Right, let's pick the brusselator, and I click it. Oops, click it. It automatically converts it to Antimony for me, and I'm ready to run the simulation. In fact. So this is a great. I find this really convenient. I mean, of course, you can do this from the command line, right? You can load in the SPML file and then export it as Antimony. You can do that, of course. But if you just want a quick thing to do, you can just do this, and then you get this is the brusselator oscillations. So that's a little handy thing. In terms of Spider as well, you have is a nice help. So, for example, if I have simulate and do Control I, nothing. Maybe I have to do that. Control I. Oh, maybe the front. Maybe do I have to put it in the front? Is that what it is? Yeah, there you go. If you put it in front, it'll also give you help in this panel here as well. Normally, it's better formatted than that, but you'll get help there. You also have a variable explorer. These are all the variables you might be currently using. This is a MATLAB. If you've ever used MATLAB, you see something similar in MATLAB. I also have my file directory where I can, load in different files. Okay, so let's get the ring oscillator, except it's in SPML, right? So that's not very handy. So I'd have to import. I could import that, for example. So and you can see here that the plots are coming down in the console, or you can have them appearing in the upper panel as well. So I've sort of got used to this. I did play with PyCharm for a little while, but then I didn't like the fact that I didn't have a console. I do a lot of interactive, I do a lot of interactive programming, interactive simulation. So I'm always, doing things at the console, asking for things like, and get the let's get the stoichiometry matrix, for example.
Veronica had a console in PyCharm somewhere.
There is some somewhere, yeah. But when I first tried it, I couldn't find it, so I sort of gave up. Yeah. The other tool you can use is Visual Studio Code. This is Visual Studio Code. Font is a bit small. This is the one. This is what I would use for doing some large-scale development of Python code. The other thing we've been experimenting with Visual Studio Code is that it has a plugin system, and we've started developing a smart modeling editor, which will help you more with the modeling building process. So
for Antimony in particular, right?
Yeah, Antimony in particular. Yeah, particularly for Antimony. Yeah. So anyway, so that's what I use. And so this actually, I'm currently working on a network viewer for Python. So that's a little example of it. So hopefully I'll have that ready by fall. So okay. How do I get out of here? Yeah, so there's lots of options for running Python, whatever suits your preferences.
Okay, I think if that's like all we mainly have to share, I think we'll take a 10-minute break, and then we'll do some reproducibility. I'll give a reproducibility talk, and then hopefully we'll have about 40 or so, 30 to 40 minutes left for comments, questions, anything anybody has, since tomorrow will be the symposium session. There won't be a lot of time to comment on like what you learned in the course, or if you have questions about your specific project. So
should I go ahead and share the survey link at this point?
Oh yeah.
Oh yeah, that would be great.
Sure, I'll do that. So we put together a little survey, and I will.
It's completely anonymous. Okay, so.
Right. All right.
Start it again. All right, so this next part is going to be a little bit of a chat about reproducibility and why it's important, and then also a little bit about what kind of tools. I know there were some questions, at least in the Slack, and probably I'm sure a lot of people have questions about other tools we could use, since most of the topics we covered talked about those that were compatible with Tellurium, but maybe your modeling project requires some special analyses that is not possible in Tellurium. So we'll talk about some other tools that might be used for various modeling tasks that have been discussed throughout the summer school. In terms of the materials for today, there are a couple of PDFs in here, which basically are from this paper that kind of summarizes some best practices that we thought were relevant to computational modeling of biological networks. And then along with that paper, there are also these resources, basically checklists to kind of guide you as you're modeling. One of which uses standards, and one is more of a general purpose approach. And then also this figure that has some modeling resources. So just take a look at that. Basically, this figure kind of goes through some different stages of modeling that you might engage in, most of which we've talked about. I think almost all of these we've talked about to some extent throughout the summer school, and it kind of it basically just links out to documentation for some tools that could be useful to you to perform those tasks. The checklists, they look like this. Again, they go through different processes you would be working on in your modeling, like data aggregation, and it goes through some tasks you might want to ensure that you perform before you're going to publish a modeling study, to make sure that you've tried to make your study as complete as possible to allow it to be reproducible by independent researchers. So this is a general purpose approach, and then the standards approach would just incorporate information about using the standard formats, and might be a little bit more rigorous in some cases. Just basically it's the kind of the highest standard. It might not always be achievable, but if you can implement some of these practices, it might make it easier for others to reproduce your work. Okay, let's talk a little bit about reproducible modeling. Why does computation or reproducibility in computational biology warrant a discussion, I guess, is the question there. Well, aside from numerous calls to address the lack of reproducibility across multiple scientific disciplines, a recent study in our domain evaluated 455 biomodels on the BioModels database, mathematical models, and found that about 50% of those were actually not reproducible, either mostly due to incorrect or missing information included in the manuscript. So that should be, if 50% of these models are already not reproducible that are being submitted and published, that's obviously a pretty big concern. But if it's not convincing to you, we'll go into a pretty fun, actually not fun, it's kind of horrifying, war over supercooled water, essentially, that illustrates the issues with reproducibility that can happen. So the story begins with these two researchers and their respective groups, but we'll kind of simplify it by just talking about the two heads of these labs. David Chandler at UC Berkeley and Pablo De Benedetti at Princeton, and of course their respective laboratories, as I said. But these two groups were basically investigating a very similar problem, both modeling the same system, attempting to try to determine what water molecules looked like when they were supercooled, but still in a liquid state, basically immediately before they would become ice. And Dr. De Benedetti's group found that liquid water could transition between a high density and a low density state without freezing, but Dr. Chandler's group simply found that there was a single state, and they didn't observe this change in their simulations. And since Dr. Chandler was more well known in the field, it was pretty difficult to oppose his findings with great success. And so the De Benedetti group basically spent a large amount of time extensively validating their code and findings. Ultimately, they requested to see the Chandler Labs code, and then over the course of 7 years, the debate remained with some back and forth critiques and further investigation. It wasn't until the end of this period that Dr. Chandler's lab finally shared their code in full. And really quickly after that happened, the De Benedetti group was able to identify that there was a discrepancy in the results that ultimately boiled down to an error in a portion of the code that provided the Chandler group with some speed advantages, but it ultimately forced the biophysical system to exist at a much higher temperature than it was believed to be in. So it completely altered the expected molecular behavior. And potentially, had they shared their code at the beginning of the debate, the error would have been identified sooner, reputations would have been spared, and most importantly, a very notable scientific result would have been communicated with confidence to the community. Of course, it's not always the case that two groups investigate the exact same problem, which means it might actually be less likely that we attempt to reproduce a study and find that it's not reproducible. So basically, ideally, we should take as many steps as we can to make sure we have extensively validated our code and made it available to ensure that it will be reproducible by others. So we'll just go through a quick overview of the practical guide to reproducible biomodeling, which follows the paper that's in the repo as well. So if you want more details, you can look at that. And then we'll look at some cultural shifts that might be able to enable reproducible biomodeling, which we can all really contribute to some extent. So first, let's go through the practical guidelines involved in making models a bit more reproducible. Like I said, it's following this paper. First, it's really important to consider the FAIR principles as sort of guiding goals when conducting reproducible research. FAIR stands for findability, accessibility, interoperability, and reuse. The principles were designed for proper management of data, but they apply to software and models as well. Ensuring that data and software can be found and accessed basically allows us to inspect published findings of other researchers. The #1 reason that studies can't be reproduced is that some critical data or software used in the original study is not shared, and the publication is not sufficient to reproduce the whole workflow. So ensuring that data and software is interoperable actually allows us to build upon previous work, which we can kind of integrate model artifacts from other researchers into our workflows. So this helps us to expand the scope of our research, and reusing existing materials ultimately saves us a lot of time, energy, and expenses, and maximizes the utility of the data and software that we're creating. So we should not try to reinvent the wheel if possible. Metadata provide, like we've talked about, provide identifier information or the context about either data sets, models, or the code. Should also follow these principles because they help us understand the model artifacts and identify them, and potentially could help us identify them as relevant to our purposes or not. So one widely adopted tool that obviously can be used to help make sure our data and code is fair would be GitHub, like we've talked about. Not only can we kind of use the version controlled aspects of the repository, but we also have a historical record of manipulations to our data and code, and it makes it easy to share our study with other researchers. So that's one resource that's available to everybody and actually does a pretty good job of implementing these principles. Okay, let's get started on the best practices for biomodeling then. So we're going to kind of go through the workflow stages or modeling workflow stages that we've talked about throughout the course. Then we'll have the recommended best practice for that task. So in this case, the best practice will be for data aggregation. And then we'll have recommended tools and standards. Obviously, the list that I have in the repo is more extensive, so we're just adding a few recommended resources here that might be maybe the best. But obviously, if your project requires something that's pretty specialized, it might be in the broader list of resources. Okay, so as we've talked about for data aggregation, we've kind of given you examples of what metadata is. Here, the best practice is that when we're aggregating and curating our data sets, we should retain its metadata and provenance. And so remember, metadata can be the conditions under which the experiment was performed and or identifier information for species in your model. And provenance then is the historical record, so how data was produced and manipulated over time, essentially, and where it originated from. Both of these sets of information kind of tell us about what's in a data set, and that can allow us to decide what's relevant to our study and how we're going to use it. So probably at some point, many of us have used Excel for storing data. As a general principle, if we use Excel, well, if you're able to read Excel, I guess, efficiently, then you could potentially you can understand the data, I guess, in Excel better than maybe in some other file formats. But if a computer can read it, it's going to be a little bit more useful. So it's important to consider ways that you can parse your data programmatically, and this will basically include retaining the relevant metadata, which is a little bit harder to do in Excel. So the recommended standard here is CPO. It's the Scientific Evidence and Provenance Information Ontology. And it's a session, basically we've talked about that ontologies are meaningful names for concepts, like a set of meaningful names or descriptions for concepts within a given scientific domain and a description of how those concepts are related. So CPO outlines a sort of standardized way to communicate about the provenance information, so the historical record of your data. And it could be useful for implementing this best practice. And then for a tool, yeah.
Yeah, is that an actual? That's not a file format, right?
No, it's not a file format. It's an ontology. So,
as far as file formats go, I know HDF5 can be annotated and is sort of Excel-like in that it can store vectors of data and stuff. So.
Yeah, that's a really good recommendation.
Yeah.
HDF5 would be a good format for this. Okay, and then bioservices we've talked about. So we've kind of, we've shown you how to use bioservices to some extent, but it's really useful here because it can programmatically search databases for relevant measurements. So you don't have to do that by hand and then copy in data and potentially leave out a lot of relevant metadata. So that's pretty useful for this task. Okay, the next rule is for model construction, and it is the practice is to make model descriptions comprehensible using structured formats and unambiguous names. So this has to do with using ontologies and a sort of common language format where parts of your model can be specified with distinct and identifiable meaning, which is what SBML is. So there are existing standard formats that help us organize our models. SPML allows us to, obviously it's compatible with a lot of different software tools. It's widely supported, and within SPML, there are distinct tags to be able to identify the species in our biochemical network, the reactions that relate those species to each other, fields for identifiers that precisely define the chemical species or the rate law, and agreed upon conventions for annotating SBML can be found in the Systems Biology Ontology, or SBO, which is one of the recommended standards here. So you'll see some SBML models basically annotated with SBO terms. And I think we showed an example of doing that as well. Okay. Then here, the recommended tool for model construction, we're gonna talk about, I already talked about, obviously, Tellurium and Antimony is a pretty good way. We've talked about how you can write annotations into Antimony to be able to add identifier information to the species in your biochemical model. And obviously you have export capabilities to SPML, so it does support the standard as well. Okay, another best practice for model construction, because it's obviously a very critical part of the modeling workflow, is to record your model construction process, which essentially has to do with generating proper documentation and also figures to demonstrate what the model system is, what assumptions were made, and all the relevant information that's required to understand your model and the design decisions that were not necessarily directly communicated from the model itself. So assumptions obviously is a really big component here to include. Okay, for the recommended standards, the Miriam standards outlines the minimal information required in the annotation of models. Obviously, we've talked about annotation. Some tools to actually perform this would be, we talked about how to use SBML utils. You can also use Antimony directly to add annotations, but libomics meta is another annotation library that can be used in Python that could be useful as well. And then another recommended standard here is SBGN. We didn't talk about this standard, but it's the systems biology graphical notation, and it can be used to basically generate network diagrams in a standardized way. Basically, it kind of gives you a common understanding of what, for example, I think on the first day of the class, we talked about different arrowheads having different meanings. And in the SBGN, all the arrowhead meanings will be consistent across anybody using that file format. So it's useful because it gives, when people are looking at a diagram, they'll interpret it the same way because it's using the standard. And I think tomorrow actually probably we might hear a little bit about visualization and how the standards support visualization from at least one of our speakers tomorrow. So you might get more of an idea about the visualization component. Okay, now I know some people were asking a lot of questions about parameter estimation. Here, the best practice is, if you are estimating parameters in your workflow, to make sure to share the estimation algorithm and ideally to perform uncertainty quantification as well. Obviously, because parameter estimation often relies on, it's not necessarily deterministic depending on the optimization algorithm you're using for parameter estimation. You need to be explicit about how you actually perform the estimation. You also should obviously share the data that you use to perform estimation or any objectives that you are using. Otherwise, it would be impossible for somebody else to perform the same parameter estimation. Here, the recommended standard is PE-tab, which you'll hear about tomorrow from one of our speakers. Until recently, there really wasn't a standard way of specifying parameter estimation routines. So PE-tab is able to is bridging the gap pretty well. So hopefully you'll get an idea for how that's used tomorrow. Some other recommended tools, obviously we heard about SBStote within this course. And for larger models, I think Herbert mentioned that PyBioNetFit's probably one of the more effective methods of performing parameter estimation. Because of the complexity of this process, it can get very computationally expensive. But PyBioNetFit does have some methods that allow it to be a little bit faster than some other options. So that is a good option. And uncertainty quantification will just give other researchers an idea of the range of parameters, basically range of parameters might fall into. It's a little more effective than just specifying one single set of parameters if there are multiple that might satisfy the or make the model represent the experimental data. So you might want to give a range of parameters, and then also that could be propagated to the outputs so that you have an idea of how effective that range of parameters are at describing the experimental data. Okay, the next practice then is simulation. Best practice is to record all simulation inputs and methods, which includes initial conditions, the integration algorithms you're using in your simulator, any random number generator algorithms, and seeds. So those last two are obviously going to be most important when you're performing stochastic simulations where you're not getting the same result every time. Again, it's basically instructing you to do as much as you can in the initial stages to explain how you explain the initial conditions you're using for your method. And of course, if any of those methods are stochastic, you're going to get different results every time you run it, which is why you would want to specify both the algorithm for the random number generator and the seed. But you also do need to specify the integration algorithm because not all integrators are going to perform the same, ultimately perform the same integration. Even within Tellurium, libRoadrunner has multiple integrators that could be used, which will give you different output, can give you different outputs. Then the recommended standard here we talked about was Setml. Obviously, it's one of the most widely accepted, and it's being improved specifically recently. So it's a good option, similar to SBML in terms of compliance with other tools. And for recommended tools, we have Tellurium, obviously we've talked about, and Composi is the most widely used simulator that is in this domain currently. So it's definitely important to note. And for some, it might be easier to use because it's GUI based for those that aren't used to programming at the command line. Okay, in terms of, in terms of storing simulation results, which I guess is kind of the next logical practice, there's also not necessarily a accepted standard for the systems biology community, but as Lucian mentioned, for data sets, HDF5 is actually a really useful format. So the best practice here is to save the structured unprocessed simulation results and share the data that's presented in graphs and tables. So for the first half of this, the unprocessed simulation results, often when you're performing your analyses on the data you've generated with your models, you may perform some statistical analyses on it. Maybe you have a massive data set and that's necessary. And in that case, you should share how you manipulated your original data set to arrive at the data you're ultimately able to share because of just file size constraints. And in the second half of this best practice, sharing the data that's presented in graphs and tables, if the only form that you're sharing your data is in a published figure, you're basically ensuring that it would be impossible for somebody else to verify, or very difficult for someone to verify that they're able to obtain the same results. It's much easier to do if you actually share your simulation results whenever possible. So the recommended tools here, NumPy and Pandas, are obviously two packages we use within Python during the course to be able to manipulate the arrays we were generating with our data sets. And Pandas actually can export easily to HDF5. I think NumPy does as well. And HDF5 allows you to add in more information about the data set. So you can sort of add annotations with HDF5, descriptions about what the data set is, titles of the data set, descriptions about what columns are in the data set. That is much more difficult to do with something like NumPy and Pandas or a CSV file. So it just kind of adds an additional level of structure on top of the data you have available. Okay, the next practice is verification and validation, which Joe touched on yesterday and today. So these cover broadly any testing that's applied to the model, the code, and the data to ensure that it is robust and a good representation of the system. And the best practice is to automate and document model verification and validation. So one way to do this would be to generate test suites that check the functionality of the code as well as the behavior of the systems that you are trying to represent. This allows you to automatically rerun all the verification and validation whenever you make changes to your model. So test suites are very efficient. And these can help ensure that any human errors that are introduced late in the modeling workflow will be caught before you actually share your work with others. So the tools here, you've seen a little bit of both of them, I think. SPML lint obviously checks SPML models for mass balance errors, and PyTest can be used for unit testing, which thereby sort of brings the elements of software or like software engineering principles into the systems biology modeling domain. And Joe showed you some examples of unit testing for some specific biological behavior. That was pretty useful. Okay, the next stage is replication. So the best practice here is to confirm that your model predictions can be reproduced in an independent computing environment. Often replication can be difficult to achieve when the modeling studies are sensitive to dependencies. So this can include the simulator that you're using, all the software libraries that are necessary to execute your simulations. Even your operating system could alter your ability to replicate replicate a modeling study. So one good check to confirm that your model can be reproduced in an independent computing environment before distributing it to others who will surely have to do so would be to either generate a virtual environment that you can use to check some of the software dependencies. When you're reproducing, when you're trying to make your study replicable, it can be useful to package it in a way that all the software dependencies are already included. So one way to do that would be like using a virtual machine like we saw for Spider potentially. If you additionally have all the packages installed that with the version specified that you need, even the Python version specified that you need or that you use to produce your original results, all of that would need to be fully specified. So the recommended tool for replication obviously is the Combine archive, because it incorporates standardized formats which can be exported and used in other simulators, which could be thought of as a sort of independent computing environment, or at least an independent simulation environment in that case. Another tool would be biosimulators, which we didn't get to talk about today, but we'll hear from tomorrow from Bilal, who's probably on the call today. Which essentially are web-based Docker containers. So they provide everything that's necessary to run the simulation for the subdomain of systems biology and modeling. So these can be a really useful tool. All right, I think this is the last stage potentially, which is packaging artifacts and documentation. And we went through this task today. The best practice here is to create packages that contain all model artifacts and documentation and deposit them in public version controlled repositories. By packaging everything together and keeping them all in a public and central location, we essentially ensure that our study is following the FAIR principles to the best of our ability. Depending on the domain you work in, there are likely many repositories that are relevant to your specific model. So try to find people in your field that are sharing their models and figure out where they keep them. But it's good to try to create some sort of centralized location. BioModels database and also Fairdom Hub are both pretty good locations for biological modeling studies. And I think Lucian shared some other resources some other model repositories that are useful. Again, using the Combine archive ensures that our study, at least for the subcellular network modeling, is interoperable because of the widespread standard support, and biosimulators will allow you to fully specify your study for your simulation study for proper replication. So all of those are also recommended tools here. And then just a comment on the version controlled repositories. By using these, you basically acknowledge that modeling does not have to end after you publish. You can kind of iterate on and improve your model, add more metadata over time to make your model a little bit more robust and identifiable, add new simulation studies to it. So just like other forms of software, your model can be versioned. And using something like GitHub is a great way to ensure that if you do update your model, those changes are tracked. Okay, so that wraps up the best practices. Now that you've built, simulated, analyzed, and validated and disseminated your model reproducibly, we're going to go through some big picture practical ideas. So one thing that we talked about today is kind of that the use of standards ultimately improves the ease of reproducibility. It makes it easier to follow the FAIR principles, and it ensures that we have tool support for the modeling studies we're engaging in. Self-contained solutions enable replication. So we talked about how easy it is for something to fail simply because you're missing some part of your code, or you're missing specification of software dependencies, and therefore somebody else might not be able to replicate your study. But creating something that's self-contained will enable replication. Model versioning supports iterative development. So again, if we're building on our models over time, it's good to have a version control implemented so that all of that is tracked and can be investigated by somebody else to see how the model is developing over time. Minimal information criteria or checklists can help you disseminate all the relevant information. So there are some standards available that kind of tell you how much information you should be including to ensure your study is reproducible. And if you can follow those, that would be very useful. And software engineering principles support clean and comprehensible code. So obviously, I think a lot of us are trying, and we've tried to illustrate throughout the summer school how software engineering can really help us as biologists and specifically as biological modelers to be a little bit more organized and methodical about how we are modeling and ensure that we're essentially making our code robust to any human error, and then also to like investigate edge cases. And one way to do that is definitely to incorporate ideas like version control, but also to incorporate testing that is extreme, that covers the full realm of our model so that our code can be validated properly. Okay, so those are the big pictures, but then the next step is how can we guide other modelers to basically follow reproducible practices, more so by influencing the broader culture. So I think this is just something that can be useful to think about. So journals, basically, perhaps you've seen this cartoon on the left floating around. I guess this was out a few months back. It was pretty popular. Everybody was posting their version of it for their scientific domain. But it basically lists the types of scientific papers we come across, including my colleague is wrong and I can finally prove it, and hey, at least we showed that this method can produce results. That's not nothing, right? Among others. This comic kind of tries to get at the idea of the poor incentive structure in academia. We've all heard the publish or perish at some point in our careers probably, and oftentimes this kind of means that the quantity of papers is prioritized over the quality, and minimal effort is often put into ensuring that our work is robust, let alone reproducible. But that's definitely changing, which is a really great sign. A lot of journals are beginning to change their approach and are offering badges on your manuscript to try to indicate the level of robustness of your study. And these badges are sort of taken from, these badges on the right are taken from the Association for Computing Machinery, but similar approaches are being used by several journals now. So if possible, it would be good for modelers to strive for reproducibility. And if there are journals that incentivize that to some extent, that's hopefully we're choosing to at least consider those journals for our publications. Next, in terms of the broader modeling community, we have community support. So obviously the Center for Reproducible Biomedical Modeling, which is putting this on, ideally is creating not only tools that support reproducible modeling, but also trying to engage with the community actively to create resources like this one, like summer schools, workshops, tutorials. We also have a seminar series to try to disseminate as much information as we can, make the tools that people are developing available to everyone that's interested in using them. So we are one center that is helping to do this, but there's also the greater Combine community, which I'm sure many people on the call have heard of, but the Combine community is responsible for developing a lot of the standards we talked about today. Their goal is to not only create a community of modelers that are all trying to seek out how to engage in best practices, but they're also creating a lot of tools that not only the standards, but tools that support the standards to actually execute the types of simulation, or sorry, yeah, I guess simulations, analyses, model building, visualization, any tasks we want to do in our modeling workflow. They're trying to develop tools that'll support that and are a great resource, and also have multiple conferences each year to share your models, share your share your software, and try to get feedback from the rest of the community, which can be extremely helpful. So in terms of big picture cultural ideas, it's just that journals obviously are starting to reward data and code sharing. So to check if studies are reproducible, hopefully this will guide the overall landscape of reproducibility in computational biology to some extent. Individuals, we know we can use open source software, open access publishing, hopefully to make our tools and also our modeling studies available for others to learn from. And communities hopefully can help collaboratively develop tools and standards that provide the functionality that we all want in our modeling studies, and a common language to communicate about our models and to develop around. Yeah, just like wanted to acknowledge those that contributed to creating this, creating the paper that a lot of this was based on, especially Arthur Goldberg, my co-author, and our other co-authors, David Nickerson, Anand Rampadarath, Jonathan Carr, and Herbert Saro, and then also members of the Saro Lab, who obviously are constantly guiding me in these sorts of studies, and from the Center for Reproducible Biomedical Modeling team and the Combine community, all of which are so heavily involved in these efforts for reproducibility. And there are some relevant references if anybody's curious and reading about the war over supercooled water from the beginning, or the study about of the BioModels database and reproducibility. I definitely recommend both of those papers. I think that's all I have on reproducibility. So I guess I can ask if there's any questions about either these practices or also the materials that are on the drive, if anyone's had a chance to look at them. If not, if anybody has questions about anything else that was mentioned throughout the course, probably some of the instructors can help to answer those. Or if you have questions about your specific modeling study that you wanted to ask, we can kind of open that up, I think, now for the rest of the period.
I guess I can start. Maybe this is now going to become a discussion.
Sure.
So first, I mean, thanks guys. So this has been a lovely summer school night. I know it's not over yet. There's still one day to go, but it's been. I really think that I didn't know about all the possibilities that Tellurium and Antimony basically give you, and I think this is really a great tool. I'm gonna say especially for teaching and for students, I see that that's really where the strengths lie, and I'm going to really take advantage of this because we are developing a new master's bioinformatics course at the University of Ljubljana, and there's going to be a systems biology, a couple of system biology courses in that, and I think this will be, I mean, this is probably what we'll use as a platform for that. So thanks for that. I mentioned this in Slack already. So we are currently we are trying to get into large scale systems biology modeling with some of our plant modeling stuff. So when I say modeling, we have large networks which are going to be about let's say 300-400 reactions large. So that's sort of the size that we're dealing with. It's plant immune signaling mostly. So and we want in the next couple of years to see how far we can get with that. And we're sort of looking for the right platform because we haven't put the model together. We have a Boolean model, but now we're slowly going from the Boolean towards a full dynamic model, which is obviously going to be super large and nothing that I've ever worked with. So we thought, yeah, maybe this is one way. One way would be to use Tellurium and Roadrunner. Another way, we've talked about the developers of PySB as well. So that was another. Both are Python based. So one of the questions, I guess, would be how much communication, how much compatibility is there between these two platforms? Does it have to go through the SBML? Do you have well, I mean, that's the first question. I have many others, but I don't want to.
Well, let's start with that one then. Yeah. So I know the author of PySB quite well now because I work with him on another project. As far as you have to go through SPML. With all these tools, because they have their own way of doing things. The one common thing is SPML, which is what it was designed for. I mean, the original reason for inventing SPML was to allow exchange between tools, even though the tools use their own formats. PySP has some nice things. It has some uncertainty quantification capabilities built in. It's quite verbose, though, when you want to write out a model. So if you want to write a model out in PySP, it takes a lot of text, unless you can exploit the rule-based side of it. But in the end, it doesn't really matter. So long as you're SPML compatible, it doesn't really matter which one you use, whichever one you find convenient. One problem with large models we've discovered is, because we have one project which has the largest model, is finding errors in those large models, which is why we're really interested in developing tools to check for stupid things that a model has done, silly mistakes and so on, because they can be so hard to uncover. So be careful with large models, and you might want to build them up in sections and make sure each section works rather than having one big, huge thing. So that's one bit of advice on large models. But we really need some new tooling to help manage large models.
One thing, SBML itself has the capability of doing hierarchical modeling, and Antimony also can help with that. There hasn't been a lot of work in that field yet, so the tools themselves, like the format is there and it's available for people. There hasn't been as much tool support for, like, robust tool support for hierarchical modeling in general. Like Tellurium just takes the hierarchical model, flattens it, and goes on with its life for Roadrunner. But I feel like, especially for a really large model, you're going to want to find the submodules, you're going to find ways to test the submodules, and then you're going to want to find hierarchical ways of stitching them back together again.
And if you do go the PySP route, check with Carlos that it's, as far as I know, it can export and import SPML, but double check with him first, because if it can't, then your model is stuck on an island, and you won't be able to move it anywhere beyond that. So
It can at least export.
Yeah. Okay.
So
yeah,
I think we can probably do both. But
yeah.
Yeah, we were actually thinking of going the other way, and setting up the model in a friendlier way with your tools.
Oh yeah, we'd definitely recommend using our tools.
Using our tools for model creation and then import them into PySP.
Well, I mean, perhaps for one or two features that maybe
right,
that maybe are yeah. So what one of the things is we really, because it's a large model, and we're gonna suffer. We're gonna have to do millions of simulations. That's what I'm gonna say.
Well, you better use Roadrunner then. That's the only option. No, really, it is actually.
Yeah.
If you're going to do large-scale modeling involving hundreds of millions of simulations, we are 10 times faster than any other simulator.
Right. Okay.
It was designed specifically for the time I knew which would come when we'd want to do lots and lots and lots of simulations.
Yeah.
And we finally reached that time now with our own projects. And PyBioNet, that optimization tool we recommend, that's one of the few we've found, well, Michael here found, that actually can do the fits properly. It depends on Roadrunner for the speed.
Okay.
So for speed, I would definitely use Roadrunner. But talking about different tools, I hop from tool to tool. I mean, one thing I like about Copaz, it has some really nice sensitivity display capabilities. And Frank Bergman did it, one of my ex-PhD students. It produces this like a cityscape of bars that go up and down, depending on the height, depends on the sensitivities. You can spin this whole thing around. And for large networks, it's a great visualization feature. And so I use Copaz for that. I mean, I don't use it for anything else, but I definitely use it for that. And there'll be other tools I might use as well. I mean, we've used another tools for comparing parameter fitting, for example. What other ones have I used? Those are the main ones I've used. So yeah, no harm in hopping around at all.
Yeah, I used to use Potter's Wheel. I'm not sure that you're aware of Potter's Wheel.
I am actually. Yeah.
I mean, right now it hasn't really been updated a lot
Right.
lately, but I really appreciated the profile likelihood.
Yes.
Which was really nice. I mean, really demanding time-wise.
Yeah.
You have to do so many optimizations,
Yeah.
but so much more robust
Yeah.
than other forms. So that was interesting.
Right. Yeah, we should relook at that one maybe. What do you have access to? A lot of computing power, because you'll need it if you're going to do profile likelihoods, fitting, MCMC, or whatever, analysis you're going to do.
Yeah, we're now in Slovenia. We're putting together a supercomputing center. No, not me, of course. So the other people working on that. So finally, we'll get some more serious access to computer power, and
okay, good.
And we have some collaboration with Oak Ridge. So yay.
Oh, all right, good. Yeah, well, we have collaboration with PNNL, so we've just been given access to their supercomputer. So Michael has anyway. So thank goodness, because we can't do it otherwise. Each optimization on one of our problems takes 36 hours.
Wow.
Yeah. So.
And that's a deterministic, or is it a stochastic?
Yep, deterministic. Yeah, it's because there's too many parameters. That's why.
Yeah.
So
I'm going to be quiet for about 10-15 seconds to give everyone else a chance to say something, and then I'll go on if no one beats me to it.
And if you want to meet, in the following weeks, just let us know.
Yeah, I've suggested a couple of dates on Slack.
Oh, right.
Early August or late August.
Late August would be good.
Yeah. Late August, then it's fine, because then I can ask other members of our team as well.
Okay, good.
That would be great. So let's keep in touch on that.
Yeah.
Okay.
Any other comments from anybody?
Issues with your own models or anything?
Hey. So my question, I guess, is, well, first, it's a great workshop, so thank you. And I'm sort of trying to process all the information that I'm getting here. And I really want to use the Antimony way of writing models, but I'm used to just writing the ODEs, and then I put them into Python and simulate them. So is there like a quick way for me to check my Antimony code so that I can feel comfortable that it's going to look like ODEs if I had just written them myself?
You can pull out the ODEs.
Okay.
I just noticed there's one little issue with it, so we're gonna have to issue there was a change in the library and the library name, and now it doesn't work properly. But there is a feature. We'll be doing a release hopefully in the next 2 or 3 weeks, but that'll fix that. But you can actually print out the resulting ODEs.
So is that with the TE utils?
Yes, but I think there's something also in Roadrunner, in Tellurium itself, actually. There's a util, TE util. Let me see, what is it? Yeah, there's a utility section in the Tellurium, which is probably under miscellaneous. Yeah, ODE extractor. Let me see if that works. Let me get a simple model here, because I got rid of the ones I just had before, so I have to create a new one. Let me just create a one-liner, okay?
Are you going to share your screen or anything?
Yeah, go on then.
Sure.
Might as well. Should crash. For this one, okay, just make it real. Okay, so there's a thing called TE utils misc ODE extractor, and it takes the SBML string. So I don't know if we did this, but to get the SBML, you just take the Roadrunner variable and ask for get SBML. Okay, cross your fingers, because I'm not sure if this is going to work or not. Oh, it did actually work. Okay, hold on then. Where is it? To string. Oh, that's it. Okay. Yeah, that's it. So that does, the Tellurium one does work. So
thank you.
Yeah, so you can have any size you want, and it'll just dump out the ODEs to you.
Thanks.
Yeah. Yeah, so one thing I like about a lot of these IDEs, they give you a lot of help. For example, I couldn't remember, right, when I started, I knew it was in Tellurium, but I couldn't remember what it was. So I hit tab. That gave me, and I thought maybe utilities. Oh, yeah, it was utilities. Then I hit the dot again, then tab. Then I thought, yeah, it's probably going to be miscellaneous. Then I hit tab again, and I saw ODE extractor. Then I couldn't remember what ODE extractor took. So I opened bracket, and it tells me SPML string. So that's the help thing like this is really handy, and I don't have to keep referring to the documentation all the time. So that's one thing I really like, actually. I can work much faster that way. I don't have to hold so much in my head.
I got a question probably accidentally asked to me personally, but so David was asking, how many software tools should you use to properly characterize the signaling pathway for a cell? I mostly use NetFlux or MATLAB, or are there other ones you suggest?
Depends what you want to do, what kind of analysis you want to do, right? I mean, if you're just going to do time course simulation, I would do use two independent tools, especially if the model is big, so that they've been implemented differently and may use different solvers, so you can confirm that you get the same result in each case. Now, there are a host of other analyses you can do, like sensitivity analysis, steady state analysis, bifurcation analysis, optimization, and there are lots of other tools that we include a lot of that, but there are other tools that do other things and so on. So it really depends on your problem. But at minimum, prior to publication, I would probably run it again on another tool if you could, just to confirm you get the same result. Anything else? There was one question on Slack about generating, synthetic networks, fake networks. They wanted to simulate small cellular networks for small world network structure. So we have this random generating tool for generating random networks, but I don't think it does. It doesn't produce a small world network. I don't think so. So all we do is we pick a random number of nodes, and then we randomly pick out of 4 types of reactions: uni, bi, bi. We pick at random one of those, and then we randomly pick the nodes and wire them up. We keep doing that until we have enough reactions. That's all we do. So we don't do anything. I've thought about it, whether it would be possible to produce large networks, because it'd have to be large to have that property. Large networks that have that property, I've thought about it, but haven't done it. But it wouldn't be too difficult, perhaps, to code your own up. So. So small world is the probability of a connection goes up. Am I right here? The probability of having a connection on a node increases if there are already connections there. Is that the idea? I can't remember now. It's been a while. Something like that. So. Any other questions? Any tooling that you think is missing? Anything you'd love to do, but you can't? Because we're also a development team as well. We also develop stuff. So one of the things that bugs me is visualization, proper visualization of networks. There's some tooling for that, but it's all over the place. There's nothing good for Python. So, but is there anything in particular tool-wise that would make your life easier other than writing the paper?
So do you have any good sampling options? I mean, for doing anything like Bayesian parameter estimation, anything like that?
Well, I've done that on toy models for teaching, and all I've used is the tooling that comes with SciPy and a couple of other packages. So I just reuse whatever Python does, and they seem to work okay. But I've only done it on small models. Those things scale really badly. Those approaches, those sampling approaches, and yeah, which is why your profile likelihood, you brought that up, is probably a better way to do it actually for large systems than trying to do sampling. You get really bogged down, I think. Those sampling methods are very weird as well. They don't necessarily start at the optimum, but sort of try to sample their way to the optimum, which can be really slow. I don't know why they don't start them at the optimum. Never quite understood that. But
Are they just searching? I mean, like if they don't know where the optimum is, that would explain it. But
that might be it, yeah. But then you could use an optimizer to try and at least give you a good starting point. Otherwise, you're just wandering over space. It could take weeks on a large model until you find, the place. But anyway, it works nicely for small systems, at least. So yeah, I just use what Python provides. What do they have? It's PyMCMC, I think, and there's a couple of others I've used.
one other person asked if we have any sort of certificate for the summer school, which I think the answer is probably no. But
we've thought about it, I mean, we could put one up.
I mean, we haven't verified anything ever, so it's hard to.
Probably just take the names here. This is the hardcore that
Okay.
stays right to the bitter end.
If you are one of the 21 participants on this call right now, you get a certificate.
I shall take a screenshot. There you go. So if we have any time in the next few weeks, we might send you a fun certificate saying you did the course, right? So that might be something. I mean, we are asked this every now and then. Actually, we were asked it last year as well. So maybe I mean, I'm okay with doing it. So especially the names I've got here. There we go.
I think Zoom also tracks who was logged in as well. So Hunter probably has that information.
Yeah. Okay. Oh, look, I'm there too. I can give myself a certificate. Great.
Perfect. Put that on your CV.
I put that on my CV.
Someone asks, can we briefly comment on other modeling approaches such as Boolean, etc.?
Well, I sort of steer away from Boolean in general, although I can see it being useful in specific cases because for signaling networks, I think it's too coarse-grained, doesn't capture enough of the subtleties. So I can imagine being used in genetic network, gene regulatory networks, where you think, genes are being switched on and off. But signaling networks are continuous machines. So
another approach? Oh, I mean, I guess there's spatial modeling.
Yeah, we don't go near that.
Which we haven't done. There's VCell, which does, and there's Zito SBML, I think, which is a newish one from Japan.
And we work with a number of the multicellular groups.
Yeah.
So if you're going to do the courses next week and the week after, you may end up writing some Antimony scripts again. Both of them use Roadrunner as the underlying simulator, again, for performance reasons, because if you've got a tumor with 20, 30, 40,000 cells, each cell running a network, that's quite a lot of computing you have to do. So you want the thing to be as fast as possible.
And then there's flux balance analysis. Flux balance analysis is when you don't have reaction rates for any of your reactions.
Yeah, we don't do that either.
We haven't done anything with that.
No.
But the basic idea is you don't have any fluxes, but or you assume and you claim that the system was set up to create glucose, say. So you say, like, what would the fluxes be if they are producing glucose at an optimal rate? within certain bounds. So you set bounds and then you say like, what's your objective function? My objective function is as much glucose as possible. Then you sort of see what the fluxes would have to be in that case. And there's usually not a unique solution to those, but there are at least some sets of solutions that would, like, if your network looks like this, it will be producing glucose a lot. I'm trying to think of other, there's a qualitative modeling. I guess that is Boolean. That's call is Bool. Boolean networks actually can be saved also in SBML and exchange between the different qualitative modeling tools. If you look at Kolomodo, I think is the group. I'm putting it in chat. Is the those are the group of tool makers who came together and like worked out how to use SPML to exchange the models they were using. So again, once you use SPML for your in your field, you suddenly have access to more tools. That's the one of the big wins of using a standard modeling format.
Yeah, okay then. Anything else? Otherwise, we see you tomorrow, I guess. What time tomorrow?
Same time.
Okay.
It'll follow the same structure. So we'll basically each hour have a new speaker, still have an hour break in between the first and second session.
Okay. All right.
So 6 speakers, 6 hours. Nice.
Okay.